Hi, good afternoon, and welcome to the ZP webinar on SensItAll. There is going to be some talking this afternoon, but I also don't want you to think that it's only talking. I'm just going to change cameras for a second, so you'll see me disappear and you'll see my hand waving around. So we are going to talk about SensItAll. So SensItAll is a platform for really commercializing ideas and technology really quickly. I've got a couple of things I'll talk about. I will talk about wearables this afternoon, but I can appreciate that most of the people who are actually either registered or attending this afternoon are not so much interested in wearables. You may be more interested in vitro diagnostics. So I will quite heavily go into in vitro diagnostics, but what I will talk about is also applicable to wearables as well. So we'll talk about microneedles. We'll talk about commercialization. So we're going to talk about how we think if people are funded correctly and they have the right mindsets that we can get them to market within 2 years. But we can sort of, be challenged and we can discuss that as we go through today. We're also going to do a demonstration. So as I said, I've got some hardware in front of me. So you won't just be hearing our voice and seeing slides. This piece of hardware that I got here, it's an in vitro diagnostics instrument. I've got some sensors as well, and we'll be doing a complete run through where we'll do a measurement and all of the data will go to the cloud as well. I also have a colleague online with me this afternoon, so you're going to see almost two perspectives. My perspective, I will do an assay and then a perspective from Guyana, and she will also do an assay as well. And we don't, I would use the word hope, but it's not going to be hope, it's actually be a fact. You're going to see how robust this really is. And you're also going to see an emphasis that Zimmer Peacock's business model has not changed over the last 10 years. We're contract developers, contract manufacturers. We've just built up more and more technology to actually speed the time to market or to reduce the time to market dramatically. So it's rather fitting that a lot of people who have signed up for today, they're either in the UK or in the USA. So we do find that the fact that I'm using this 2-year manifesto and that sort of that political resonance, we're not standing for Parliament or for president election this year, but we do have a 2-year manifesto, and the 2-year manifesto is we think we can get people to market much quicker than historically has been the case. Now we think we can do it through the SensItAll platform. So I have at Zimmer Peacock, we have a whole series of technologies that interconnect. So if you're looking at my camera, I'm holding up my phone. My phone has an app on it. This app is part of the technology. We also have sensors or screen printed electrodes that I'm also holding up at this point. And then I also have in front of me the hardware as well, which is the SensItAll platform. So all, let's say, come together, but also cloud technology as well, that this piece of technology that we're going to run this afternoon will also connect to the cloud. So all the elements that make up a modern in vitro diagnostic system of sensors, electronics, apps, cloud connectivity will all be demonstrated on one platform this afternoon. So that will be SensItAll, but what we talk about is equally applicable to wearables as well. So we have wearables and we have IVD. If you've got a choice in life, always go on the IVD in vitro diagnostic direction first, because wearables is a really tough technology. I would almost say you need 10 times as much funding to do a wearable. So for example, you were making a glucose sensor for diabetics, you'd end up spending easily 10 times as much to get a wearable CGM, a wearable continuous glucose monitor, as opposed to an in vitro diagnostic. Now this is, you will get a recording of this. So when I speak fast and things flash up in front of you, and you haven't got the time to respond, don't worry, because you can watch the recording in a bit. But when you do watch that recording, or if you're watching on a screen and if you use an Android phone, this QR code will come up again in a bit. But this QR code is scannable off an Android phone. And if you scan it, it will guide you to install an app called SensItAll. So the first part of one of the parts of technology development and biosensor development is, where's the software? Well, the software is available through this QR code, and it's already on the Google Play Store. So you can get a sense of, let's say, software deployment is actually through one of the most friendly user interfaces, which is the Google Play Store. especially as we do have quite a few people in California that use our technology. We are actually on Apple as well. So if you're using an iPhone, you can scan this QR code, and that will take you to the Apple App Store, and you will find again a piece of software called SensItAll. So this is really a 2024 kind of technology, because I'm going to show you some hardware, but it's actually driven by an app on my smart device. And the app is a great, let's say the smart device is a great bridge, because this app is going to connect the hardware and what the hardware does to the cloud. So you can, any kind of, let's say somebody developing an IVD today, an in vitro diagnostic today, I think should have cloud built into their technology offering, because then you have the possibility of making revenue through the sensor. You have a possibility of making revenue through the electronics or the meter, and you have possibility of making revenue through the data as well. So sort of data as a service. So 3 routes to revenue. This used to be called the razor blade model, where you would sell meters and you would sell sensors. That was only a 2 revenue model. This is a 3 revenue model. Anyway, that said, you can install the app on your iPhone. Now, it's not, you will see that we can do research on this, and we can also do real applications on this. So today, Zimmer Peacock uses this SensItAll hardware for helping in the food industry. So we've got a couple of food analytes here. pH is also something that we can measure on this platform. pH, you can measure pH of blood, you can measure the pH of food, you can measure the pH of water. pH is a really important, let's say, analyte. Many people in this world are interested in nitrates. If you look at the River Wye, the River Wye's got a particular phosphate problem in the UK, but nitrate pollution, Poole Harbour, for example, has a lot of nitrate in it. So nitrates is a real problem in agriculture, in aquaculture, and nitrate is also something we can detect on this platform. Total antioxidative capacity. Just out of interest, if you end up installing this app on your phone and you then follow this video and you scan these QR codes, you're going to find that you can keep going to these different analytes. So you're suddenly understanding that actually this is not analyte specific. Every time these guys generate a new QR code, we can change the target for what that hardware can do. And that's exactly what it can do. So glucose, potassium, for example. Now, if you're in the startup world, you may be dealing with academic scientists, or you've got your own scientists, but your scientists will be using electrochemical methods called amperometry, open circuit potential, electrochemical impedance spectroscopy, linear sweep voltammetry, cyclovoltammetry, square wave voltammetry. Each one of these QR codes will make the app, rather than being an analyte specific app, it will then become a research tool, so it can do amperometry. So if you're doing glucose sensors, it'll be amperometry. If you're doing what's called ion selective electrodes for potassium or sodium or pH, then your scientists will want to be doing open circuit potential. If you're trying to do immunoassays for sepsis or for some sort of binding assay, then your scientists may have talked to you about electrochemical impedance spectroscopy. They may have talked to you about linear sweep voltammetry, cyclovoltammetry, or even square wave voltammetry. So this is a device that the app is in Bluetooth connection with the hardware. So when I say that, I'll just change screens just really quick. So the meter that's in front of me does not know what to do unless it's being told by the app. The app is in Bluetooth connection with the meter. And how do we tell the app what to do? We use QR codes. And you're going to see very quickly that actually there's a QR code which is designed so that clients can actually use this technology as their product. And the whole business model at Zimmer Peacock is, despite reputation, we actually don't want to be super famous. We actually want to be quietly in the background, allowing other companies to actually go forward and develop the technologies that they're interested in. As I say, this is really a webinar this afternoon about IVD, in vitro diagnostics, but just for interest, at Zimmer Peacock, we also do wearables, wearable microneedles, and I will talk about that. It's actually, this is our 10th year, so if people don't know us too well, we're actually been around for 10 years at this point, and we've been ISO 13485 for approximately the last 5 years, and we make most of our revenue from development and manufacture and we make standard products as well. I'm not going to spend a lot of time on wearables this afternoon, but just to say that at Zimmer Peacock, we do make sensors that go through the skin. We make sensors that lightly go through the skin using microneedles. We make sensors that sit on the skin. And we also make sensors that get embedded within. And when I say embedded, I mean, maybe the word is better, implanted. But so far, we've worked on fish and pigs. Just to give you a sense of what's the power of electrochemistry, the power of electrochemistry is this package is something that I'll talk about in a minute. But this is something that gets implanted into fish. The fish are not killed during the insertion. The fish are actually returned to the, I want to call it a fish tank, but it's really a sort of, this is really salmon farming. So there's more than a tank. It's a sort of a holding. But just to give you a sense that that's 15 millimeters across and 38 millimeters long. So 15 millimeters, approximately 3/4 of an inch. And the electronics is just this little tiny board in the middle of this. So that gives you a sense of the size of which the electronics to drive these kind of biosensors, how small they actually are these days. As I say, this actual sensor package ends up getting embedded in fish. This fish is not dead; it's anesthetized, and this is the fish after that implantation, and they survive the operation. What we're doing is, if you look at Zimmer Peacock, despite the accents, we are actually in Norway as well, and we're helping with the salmon farming in Norway through implanted sensors. This is a continuous glucose monitoring signal from a fish. This is after 9 hours, we fed the fish and the glucose sort of jumps up. So we do have technology for people who are otherwise interested in wearable sensors, but I don't feel like that is the tone that I want to take this afternoon. But if you are interested in wearables, just know that Zimmer Peacock is capable of doing wearables as well. What I am going to do now is talk about the problem that I perceive. What you're going to find out is that at Zimmer Peacock, we do technology, but the technology is not random. It's actually driven by a philosophy, and the philosophy is always been, almost day one on our website, we wrote that Zimmer Peacock exists in part to commercialize technology that's coming out of academia. That line has been on our website for 10 years now, and it's matured in our understanding of what that really means. But actually, the philosophy has always stayed the same. So let's talk about commercialization. There's something called the Valley of Death. If you're in a startup world or you talk to academics, the Valley of Death is a problem where you've got technology readiness levels. So if you first talk to somebody about an idea, that might be technology readiness level number one. An academic paper where they've gone in the lab and they've done some testing with an analyte and they sort of show you a signal. They're bringing the technology at that point to technology readiness level number 3. Now, the whole idea of industry and startups is that we're going to somehow bring them to technology readiness level number 9. I say somehow. I'm going to show you how I think it can be how that can happen. So the job of the commercial sector is to, in the end, these are for-profit endeavors, but we're trying to, bring great ideas that we think can make for great businesses out of academia and put them onto the market. Now, there's this gap between where academia finishes and where industry wants to get to, and everyone, well, not everyone, but academics sometimes call this the Valley of Death. Now, governments try to span this Valley of Death. They try to span it by bringing in the US, we have SBIR grants, and in the UK, we have, for example, Innovate UK grants. And these are all planned to kind of help get technology from technology readiness level number 3 to higher technology readiness level so they can actually get funded. This Valley of Death has been talked about since I've been in industry. So this is over 20 years this has been talked about. And do I think that this problem's been solved? No, I don't. I don't generally think that governments are really keen in solving it. But is it really solved? I think the answer is no. And I actually think that we need to relook at this problem. So there's a philosophy here, which is we want to get people onto the market quicker than is typically taken with a higher success rate. The problem we have is this valley between where academia finishes and industry needs to get to. And so we actually just want to close that gap, literally close the gap. So it's not such a big gap, because I think what's going on here is that the way people do academic research and early development. They do it on instruments and lab equipment that looks nothing like the product. Now I'm going to quickly I'm going to quickly change screens again, and I know this is very small on your screen, but the technology that can be used for research, which my hand is touching upon here, is exactly the same technology that can also be used to get to market. So when I say we're trying to close the gap, we are literally closing the gap, because actually the development that you would otherwise need to do in order to translate that academic research into a product just went away. But I'm going to expand upon that during the talk today. Now we have a physical implementation of this closing the gap, and we call it the SensItAll platform. You'll see it in a minute. It uses everything at ZP is designed to work together. So we make really excellent screen printed electrodes, because it's part of the problem that people can't make reproducible electrodes, therefore they can't make reproducible sensors. Zimmer Peacock, we do a really good job of doing that. If you watch this video later on, or you're watching a recording, scan this QR code, install the app on your phone, scan this QR code, because this will make the point for me. If somebody's in a lab, they may do an experiment which is called open circuit potential. But if that open circuit potential method goes into the real world, they may rephrase it as pH, because pH is the science behind pH measurements is open circuit potential. Suddenly, the same piece of hardware can do the research by scanning the QR code on the left, and then it can be the product by scanning the QR code on the right. So you go from research to product, as long as you can operate a smartphone, which I think even most people can at this moment in time. So we're trying to close that. We're trying to get rid of this valley of death. Let's expand this problem a little bit more. Right. If we look at the spend versus time profile for a startup, most startups will tell people, yeah, we're going to get to market in 5 to 10 years. I think realistically, people say it's going to take 5 years. I think it often takes them closer to 10 years. And actually, this can also go out to 15 years. So in reality, it takes longer than people otherwise want it to take. And the success rate for anyone actually bringing a technology to market, the failure rate is 90%. So this crossing the valley of death problem has in the end a 10% success rate associated with it, and that's really only getting to market. That's not staying on market and being viable for several years. Now, if we're going to reduce this 5 to 15 years that I'm talking about to 2 years, something has to give. So along the bottom here, I've labeled up basic principles, which is TRL1, technology concept TRL2, proof of concept TRL3, up to system deployed TRL9. So these are checkboxes that if you achieve them, especially in a sequential manner, then you've got a technology that's kind of probably on the market by the time you get to 9 here. So I want to condense 5 years down to 2 years. You can't just do it by doing everything faster, but actually we want to eliminate things. Basic principles. Here's a basic principle, making electrodes. People spend a lot of time just going around in a fishbowl making, different electrodes. So, we through this system are trying to kind of cross some of this off. Technology concept. This concept is, we'll do a demonstration on a system in a minute, and that will be the concept. Proof of concept. I'll do a demo in a minute on a piece of technology, and the proof of concept will be something that you will do. But system prototype, system complete and qualified, and system deployed. So, for example, this week, with the SensItAll platform, today is Thursday, we've shipped 2 this week. By the way, we've shipped a lot more, but I'm just saying to you, this system is already deployed. It's already making its way out there. You can imagine, actually, most startups spend the first 2 years just building up the hardware platform, but this is a platform that's already being deployed. But it does leave you with proof of concept to do. So if you've got a new protein or a new immunoassay that's being developed, that has to be developed. You guys still have to go in the lab. You have to test with relevant samples, and you probably have to do demos. But let's say there were 9 checkboxes here, and I'm going to condense everything down from like 5 years to 2 years. Well, I'm sort of saying I'm eliminating 5 of those check boxes here and just leaving you with a subset. So when I make this statement, I think we can get you to market quicker than you otherwise would have. It's not because we did everything quicker, it's because actually we eliminated things. And that's how we can sort of make this claim. So we got a platform, we call it SensItAll. It's an OEM solution, so it's an original equipment manufacturing solution. So it's inherent at Zimmer Peacock that everything we do is contract development, contract manufacturing. It's just we built more and more of the hardware and technology now so that it's not reinvented every time. Every time, how many times do we see people reinventing things? I'm sure, it's a frustration for everyone. But we have this hardware, we have the electrodes, we have the app. Don't forget you can install the app after watching this video. We have a cloud system. You're going to see that cloud system used live. It also has what we call an API, an application program interface. So your third revenue stream, I said, would be that data as a service. So whoever your customer is going to be, for example, let's say your target in the end is the NHS, or it could be hospitals in the USA. They will have electronic medical records. How are you going to plug into the electronic medical records? You're going to plug into the electronic medical records using APIs, application program interface. This is already built into this system. So we have hardware, we have electrodes, we have apps, we have cloud systems, and we have API built in as well. So let's go nice and fast. Now I want to say here, there is some value add that has to come from you, your startup, and your academic researchers. And that's what happens on this electrode. That's the value add there. See, there's not value add in making new hardware. You can always find mechanical electronic engineers, and engineering is systematic, if I can say that, or deterministic. Actually, I should say deterministic. If you pay people to do something, they generally, in engineering, they can generally do it. Give me a box, give me electronics, they can do it for you. But when you pay people to do science, you suddenly find that it takes a lot longer and it's a lot harder than anyone ever thought. Because here you're trying to deal with molecules, and then trying to make molecules go in the right direction is very different from trying to make a square, trying to make a box. So I'm just saying that engineering is deterministic. You really need to add value, which is actually doing the assay itself. So we say, actually, and we are biased. I would like people to use our screen printed electrodes because our screen printed electrodes work on our SensItAll platform, and we are biased. And we want people to functionalize the surface. So I think the real value add is, what is that biochemistry that you've got your scientists developing? Whatever they're developing, that needs to go onto the electrodes. So that I would call that the assay. That's a good place to spend your time and your money. How are they going to analyze that signal? They're probably going to use EIS, that's electrochemical impedance spectroscopy, maybe square wave voltammetry. So they're wise to spend some time on the method. And then there's the bit that really very few people actually get to, which is actually the calibration part. How am I going to turn raw signal, squiggly lines, let's say, into a number that's meaningful to a clinician? That's the calibration. Now, the SensItAll platform will allow us to, if you develop the assay, you develop the method file, and you develop the calibration routine, we can program, well, we can apply the assay to the electrodes, and we can apply the method file and calibration to the platform. And so suddenly you're only developing 3 things. Only is a bit of a term: the assay, the method, and the calibration. And then suddenly you're able to actually launch that onto a robust product platform. So your path today is, we do have the electronics, we do have the hardware. I put this picture in here because somebody sent me this picture of the screen printed electrode and asked me could I support them. I was like, no, it's horrible. We do make some nice screen printed electrodes, and even today, if you said to us, "Oh, I really want my own branding on this," no issues. We can just cover these up with nice stickers and get rid of ZP. So ZP does not need to be anywhere in your product offering, especially when you're trying to talk to investors. We understand that scenario. You want to have people demonstrating your technology so that investors understand the values coming from your company. So we want you to focus on that, which is doing the assay, doing the method file, and doing the calibration. So I showed this QR code earlier on, but just repetition. If you scan this, you will find that the app is already deployed on the Android Play Store. If you scan this QR code, you'll find that the app is already deployed on the Apple App Store, and it is hard to get apps onto the Apple iStore. You have to meet some. Apple is very stringent on its quality. You know, that's why Apple things work, because they're quite well, they are stringent on quality. Now, you can then, especially if you're the kind of commercial lead for your company, get your scientists to put that app onto their phone and then to scan this QR code here, because then they're going to suddenly find out that actually they can do amperometry. Get them to scan this code, then they find out that they could do open circuit potentiometry. Then get them to scan this, then they could find they could do EIS, electrochemical impedance spectroscopy, linear sweep voltammetry. Just get them to essentially do their own demo, cyclic voltammetry and square wave voltammetry. So suddenly they're going to understand that this app can do the fundamental science that they want to do, and then get them to so what it will do is it will change the way the app looks every time, but actually also what the app can do every time as well. So there's a sort of, be very clear to them that the app is actually changing its functionality. And then get them to scan these QR codes, because every time they scan these QR codes, they're going to start to realize that actually I can go from a research experiment to a analyte very quickly. These guys can just give me a new QR code, and actually I'm starting to get towards that user experience. I think the biggest gap between TRL, let's say 3 and TRL 5, Technology Res 5, is this user experience. But this is what this platform is really good at doing, is really narrowing the gap between the lab experience and the user experience. So I do want them to scan those QR codes and to follow this. Now, scan this QR code. What this QR code will do is it will actually bring up a screen that is blank on your phone. Understand that everything that is showing up in a red box, from the name to the logo to the colors to the name here, everything can be changed. So it doesn't have to say Zimmer and Peacock. It can say Newco, whatever your company is called, wherever your assay is. If it's sepsis, for example, it could be the sepsis name for example. So that QR code already exists, and it's to demonstrate to you that we almost have 3 bins of QR codes: R&D QR codes, application QR codes, and then your QR code. Your QR code is the one that will actually make the app and the system become Newco, or whatever your company's actually called. Now, the only reason I've got this video up here as well is I want to talk about user experience. There's a big gap between research that happens at TRL3 and research, or the ease of use, that happens at TRL 57, 567, let's say. But this wasn't something that was rehearsed, but actually I got a couple of young volunteers actually to run this SensItAll platform. And admittedly, they most of the time they actually hear what's going on just because they sit in the background. But they ran the whole assay without any kind of, let's say, prompting, and actually got the whole thing to work. So I'm really, really sorry, but this platform is child's play, and it literally is child's play at this point. Now you'll see me do that child's play in a bit anyway. But so QR codes read by the app. Every time a QR code is read, the app changes. The app at that point has been programmed. So whatever you develop in the lab, we can turn it into a QR code, and we can then program the app through that QR code. The app will then tell the hardware what to do. And every time we scan a QR code, and there has to be the sensor as well. So sensor and QR code effectively go together. So you could think of it like this. There's only 2 variables. What is that assay that you're developing, and what's that method file? And that method file will also have a calibration associated with it. But we can tell the software immediately what to do through the QR code. You're not even programming. So programming and many of the engineering functions actually go out the window. So QR codes drive the app drives the meter, and the meter then drives the sensor, and then the app will get the data. The meter will give the data back to the app, and the app will actually end up sending the data to the cloud. But I will demonstrate that. So now even the cloud is something that we started 10 years ago. Sort of everyone was talking about big data, so I thought, well, that sounds interesting. We better get into big data. we need some sort of cloud system running here. So we have a cloud system called Julie. So I will demonstrate that in a minute. But it's just to say it's really important. If you're doing product development and you're going to be having clinical trials, you really want to see the data coming in from those clinical trials in real time. And the fact that all the raw data immediately goes to the cloud is really powerful for you. So you'll see it during the demonstration that the data will appear on the phone. So the clinician will get data on their phone. They'll be happy because they can, write it down or they can make a diagnosis. And your scientist will be really happy because the raw signal behind that number also goes to the cloud as well. And the cloud will let you easily share with your, you don't have to download files, you can just share it through the cloud as well. So I'm going to do a SensItAll demonstration. So what I'm going to do is I'm going to stop sharing this for a second. I'm going to change to a slightly different, I'm going to change to a different camera. So this is the SensItAll platform. I'll do an assay now. I'm going to, first of all, note this. I'm going to click this down. That clicking down means I can take this sensor out. You will get a better demonstration from Guyana, but I'm going to take one of these sensors, take it off, put it on. What I'm going to now do is I'm going to bring my phone into shot. So my phone, which is here, is in Bluetooth connection with this device. I'm just going to change this name slightly. Now that name is important because that name will allow me to find that data later on in the cloud. Now I'm going to take the sample. I might just change screens for a minute. Why do I like electrochemistry? Because this is a sample that I'm going to testing. Like how horrible and dirty that sample is, but electrochemistry is one of the few sciences, let's say, that can deal with real world samples. So I will be taking, and another thing about electrochemistry that's really good is it can deal with small samples. So this is 50 microliters. You'll struggle to see me put the sample on here, but I'll put the sample on. 50 microliters has gone on there. That's more like a drop of blood. And now what I'm going to do is I'm going to go to assay. I'm going to go start measurement. Guyana will show you later, but if I put my finger here, you can see my finger slightly pulsing with light. That's because we've got some really nice LEDs along the front of here, which are flashing away. And they're telling me, let's say I was the nurse or the doctor or the patient, oh, the unit's working because the lights are flashing. It gives people that user experience, that confirmation that things are working. You also get a progress bar here. Notice there's no raw data being shown here because, real people don't really want to see data. And I've got here the results in parts per million, 410 parts per million. I'm just going to change cameras for a second because I want to we've had a bit of a celebrity visit this afternoon. The celebrity visit is this. I know I was testing this product, and I know you might say, well, what's this got to do? But the reason this is, if you listen to Radio 4 in the UK, they were talking about some samples in Denmark, which were too spicy and hot for people to eat. So we actually got some of this in off Amazon, and I've just been testing it. It's actually not that spicy and not that hot. So unfortunately, it does say something about the Danish taste. But what I'm going to do is I'm going to share my screen with you because, in fact, first of all, I'm going to hold this up and say, this app does say something to me. This app gives me the results and it says upload complete. What does upload complete mean? Well, let me go to this screen here. This is Julie. It's not trivial to build a cloud system like this, but if I just refresh, like that, then I can actually see my data. There it is there, just popped up now. So I'm loading it up. So this is essentially going in real time from my, and here it is, beautiful. Here's my results, great. You do not have to show this to nurses or doctors. This is for your scientists so that your scientist in real time can tell whether it's working. And it's really smart because there's the result that the user will have seen. And here is the raw data. So your scientist will be familiar with raw data because they will have been gathering it in the lab. And so they'll be able to at a glance say, does that look right? So that's the first kind of quality control. And also they can program how the data is actually processed. If I click on here, suddenly, I know this is a lot of, but we can do things like smoothing of data. We can do area under curve calculations from data. We can do formula fitting. So suddenly all the things that would otherwise would the exercise in turning their raw signal into meaningful numbers can all take place in this cloud system. And I'm obviously super biased, but it's pretty powerful that actually suddenly things that you otherwise done in the lab, it's much more easy, let's say, to have converted it into a product. Now, I gave you my perspective on the SensItAll. Now, what I'm going to do is I'm going to mute my mic. I'm going to turn off my camera, and my colleague Guyana is going to come online. And Guyana's one of our engineers, and she'll give you a demonstration and also a slightly different perspective as well. Okay, thanks, Guyana.
Thank you, Martin. Hi, everyone. I'm Guyana. I'm an engineer here in Simon Peacock, and I'll show you what I have with me right now. I have the SensItAll device. I have a QR code, which is for ginger assay particularly. I have some sample to test, and I also have some ginger sensors. I also have a mobile app with me. Right now it's Android, but you can also download the iOS version from the Apple Store. The device is really small. It fits in a palm. It has USB-C connection. We can connect it to the power source. Right now, it's connected to my laptop. So maybe we can start. For that, I will press this button for 2 seconds. Now I see that LED is on, and I also see the status bar here. It's gliding blue light. It means the device is ready to be connected to a mobile phone. So I'll show what I have on the mobile phone. So we can try to connect to the device. So for that, I will press the Bluetooth icon. And now it's searching for available devices. And now I see that SIA 2037 is available. So I will click on it, and then it will initiate the connection and change the screen to the home screen again. And after that, we will scan the QR code. For that, I will press the icon, and I will hover it on top of the QR code. Yeah, and we see that the header color is changed, and we see the ginger sensor, and we see how many steps do we need to finish this assay. So in this particular case, it's just one step. We can also change some test settings. I will click on the card, and here I can say the sample ID. I will name it ginger test 2. And the cluster name, I will call it demo. So you can think of the cluster as a folder name. So these two parameters will make it easy to find the data in Julie later. And operator ID, I'll just keep my name as it is. Now I'll press go back button and then go to assay to see the instructions. So I have a ginger shot solution with me, which I previously mixed with a ginger buffer with 1:10 ratio. And here is the solution that I prepared. So because it's the 1:10 ratio, I don't need to change anything on the mobile app. But I also have options to choose 100, 1,000, and 10,000. After this, I will need to put the cartridge. And then I will click this moving part up. And then I can see that the connector pads from the sensor are connected to spring-loaded connectors from the device. Yeah, now I will move back. And I will go to assay. I just wanted to verify that Bluetooth, that the device was connected. I just heard disconnecting sound. Okay, I will go to assay again. So we placed and locked the cartridge. Now we will need to pipette some solution on top of it. Yeah, usually 50 or 40 microliter is enough to cover all electrodes. This is done, and I will press start measurement to initiate the measurement. So right now what is happening, the device is running cyclic voltammogram and sends the data to the mobile app, and mobile apps gathers this data and then posts in Julie where our data is stored and also analyzed. And once Julie finished the analyzing, it sends the results back to the mobile screen. And this whole process takes 30 seconds in total. Yeah, and now the results are back. We see the results with molarity and also milligram per liter. I will remove the sensor and also show that the LED is green. That means that data is uploaded in Julie. If I change my screen to Julie, I'm in demo cluster already, so I don't need to change that. I will go to SIA 2037, which is the device that I currently using. I'm clicking on it, and here I should see Ginger Test 2 data. So when I click on this one, now I see the raw data, I see the first scan, and if I go a little bit down, I see the second scan, and I see the pre-processed data. I see that we are using one of these peaks to determine the molarity of the ginger in the solution. Yeah, I can also see the calculated peak height and some more results in this tab. That's all I prepared, Martin.
Thank you very much. I appreciate that. So we like to be efficient with people's time. So I just want to kind of do a quick summary here. So, we did talk about microneedles, but it's because some people are actually interested in wearables, and that's something that you can kind of reach out to us on. But we have actually focused on really IVD this afternoon, vitro diagnostics. What you can see is that we actually have a very robust platform that we can just turn up, do these live demonstrations. I did one, Guyana did one, and we did the measurement. Nothing crashed. It all went to the clouds. We could see our raw data up there, and so we were able to perform those kind of demonstrations. So the whole mindset at Zimmer Peacock is if we can somehow work with people and they can get onto as much of our technology stack as possible, then really they can get to market and to powerful demonstrations much earlier than they otherwise thought. And we can overcome this problem of this valley of death between TRL3 and where people really need to get to. So if you've got any questions on ZP, then don't hesitate to reach out to us. I obviously recognize the attendees here this afternoon, so diolch yn fawr. And we will speak to some of you soon. But don't worry, I'm going to send out a recording very shortly, so you can watch it, you can play with the QR codes, you can also share it with the other members of the team as well. So thanks, Guyana, and have a good rest of the evening for the people attending. And if you're watching from California, have a good rest of the day. Okay, thanks very much. Take care. Bye-bye.